We're back. Come on, Mark. Come on, Richard. Turn your cameras on. Don't leave me here all on my own. Look, we could have got Mark. You're in the lab and everything. We could have got you to move your camera around, show us what goes on.
Yeah, I thought it'd be an interesting location to
could have done a lab tour. In this session, I'm joined by Dr. Mark Dallas, who's an associate professor in cellular neuroscience at the University of Reading, and Dr. Richard Lofthouse, who is a research fellow at the University of Aberdeen, and who also does some work with a company called TauRx. Hello, both. Thank you for joining.
Yeah, good afternoon.
So we're 3.5 hours in, and we're doing okay, I think. Thank you again. And while I go find something else to eat, I'm going to ask you to do your introductions. Mark, why don't you go first?
Yeah, sure. Thanks again for the invitation, Adam. And yes, 3 hours in, and you're still kind of cracking on, full of verve. So great to see. So yeah, our research fundamentally is looking at the different cell types in the brain. So as we all, I'm sure, aware that Alzheimer's disease, dementia, is a neurodegenerative disease. So that's the nerve cells that are dying. But a lot of research that's been funded by some of the charities that you're supporting today, Adam, has pointed the finger at some of the other cells in the brain. So typically termed glial cells, these are once thought of as the kind of support cells. They were really thought of just there for structural support. But some of the exciting research that's come out of labs across the world has indicated that they are involved in the disease processes that underpin dementia. So our attention is turned to looking at those cell types. So we'll maybe talk a little bit more about microglia, you might have heard of that today, and also astrocytes. So two different cell types that are in our brain that are really important in not only setting the brain health, but also can have an impact in terms of the pathology and the disease progression. So we are actually starting to look at the function of these cells in isolation to understand what only do they do in normal healthy brains, but actually what goes on in terms of people living with dementia and how do these cells change and not only contribute to that disease, but also may actually give us a little bit more of an insight into some of those really early changes that happen in terms of the brains before someone actually goes into the clinic and in essence goes through those kind of memory tests and gets that diagnosis. So really there's kind of two strands, is thinking about the function of these cells, but also thinking about the opportunities that exist in a diagnostic world as well.
And this is with a view then that you can use that to counteract whatever is going wrong.
Yeah, exactly. So by understanding the kind of basic mechanisms, whether it's a protein or a target or a receptor within these cells, we can look to see if there's drugs readily available. So there might be drugs already on the market that actually are used to target some of these receptors that we've found in our research, and they could be repurposed for dementia. So it's about offering opportunities to kind of reset the function of these cells that we've seen actually, in essence, becomes perturbed or becomes compromised in the disease process.
Fascinating. Thank you, Mark. Richard, tell us about introduce yourself. Tell us about your work.
So, hi everyone, and pleased to see you again, Adam, and nice to meet you, Mark. My name is Richard Lofthouse, and I'm a research fellow up at the University of Aberdeen, and I'm postdocing now. I just finished earlier in the year my PhD my work is purely looking at the tau protein. So you might have heard of these pathologic hallmarks of Alzheimer's disease before, very commonly referred to as either beta amyloid or tau protein. And in our lab, we're making antibodies against the tau protein, and with the aims of detecting new biomarkers. I'm sure that's a term that's been said many times so far in your 3.5 hours.
We have heard biomarkers a lot, but we're moving on now. It's the first time we're going to talk about amyloid and tau, though, and microglia today in this session.
And also with the hope of some therapeutic benefit further down the line using the antibodies to target the tau pathology. That's really we're very focused on that tau protein.
Thank you. So, Richard, we often hear about amyloid. In fact, anybody who's interested in dementia can't have really avoided. In fact, I had my hair cut especially for yesterday, and even the hairdresser said to me yesterday morning, "So I hear you've had some good news this week." In the news, she knows I work in dementia. And of course, what you'll have heard about a lot in the news this week is about amyloid and stickiness and how this clumps together and proteins and things like that. What does that actually have to do? So what's tau then? What's that all about?
So tau is...
Well, do you want to start, actually? Why don't you give a better little quick rundown as to, actually, no, Mark, you tell us what amyloid is, and then Richard, you're going to tell us what tau is. Go on, Mark, what's amyloid?
Yeah, so amyloid, is kind of a bit of a buzzword.
Put you on the spot there, sorry.
Yeah, no, amyloid's it's a bit of a
question list.
Yeah, it's a bit of a buzzword in terms of the media, and that's something that's often focused on in terms of when we talk about dementia. So we know that the amyloid is a protein that is critically involved in the disease process. So that leads to the accumulation of the plaque. So way back when Dr. Alzheimer started to look at post-mortem tissue to try to characterize the disease, he saw these peculiar substances in the post-mortem brain. So looking at brain slices of people who had presented with symptoms that we now know are associated with dementia and Alzheimer's disease. And he noticed that there were these large plaques that accumulated in the brain. Research has progressed. We've started to understand what is the components of that particular plaque, and that is the amyloid protein. So the amyloid protein, we all make it, but in terms of those that are living with dementia, there seems to be an overaccumulation of that particular peptide that leads to, in essence, its amalgamation and that sticky composition that leads to the plaque. So that's why a lot of kind of the therapies, and particularly the ones that have been in the news recently, are targeting that protein. If we can look to try and, in essence, change the course of that amyloid peptide aggregation to form those plaques, we might have a handle, and we might have, in essence, a therapeutic opportunity to try and alleviate some of the symptoms that we see with those living with dementia. So it's one of the characteristic hallmarks of the disease, but I'm sure Richard will also attribute to the value of tau as well. So that's one of the in essence challenges of Alzheimer's disease, is there's multifactorial processes occurring. To focus on one might be to miss the overall presentation of an individual living with dementia.
So, Richard, thank you, Mark. What about tau? What's tau got to do with it?
That was a great overview of sort of amyloid, and I think it's important, just as you said, to focus purely on one. You would miss maybe some important aspects of the hallmarks of the other protein. So tau protein itself, I like to think of it as scaffolding for neurons almost. So it's inside the neuron. It provides structural stability. It helps transporting waste products away from the synapse, which is where the important bit, or some of the important functions of the neuron, take place. And it also transports nutrients down there as well. And for whatever reason in Alzheimer's disease, tau protein begins to self-aggregate. So it sticks to itself and forms a very stereotypical structure called a paired helical filament. These progress, and eventually neurons die, and the pathologic tau is released into the intracellular space. And this sort of leads to progression through associated and connected neurons. And what's interesting about tau is that the areas where tau first arises are thought to be some of the first areas affected by Alzheimer's disease. And you can track the spread of these tau tangles, as they're referred to, throughout the brain as the disease worsens. And so therefore, tau could be a really useful therapeutic target in itself if it's closely related to the disease progression. If you can prevent tau spreading, you could hopefully prevent the disease getting worse. Yeah.
And so is that one of the, of course, anybody who's watching who isn't in the kind of research community will have taken the news that they've had this week a bit like my hairdresser did and went, oh, well, this is great, then there's a new treatment. But actually, the controversy is that it's more complicated than that, isn't it? That you're probably, a single treatment just for amyloid might have some benefit, but the chances are you're going to have to do other things as well, which would include addressing tau.
Yeah, I mean, I think that's kind of one of the kind of caveats that always, as a scientist, you always rely on, is it's more complicated than that. But I think that the results certainly were, in essence, a really kind of a line in the sand to say that actually targeting the amyloid can have an effect. I mean, the effect was quite small, but it is an actual change in terms of the disease progression. Think about now bringing in kind of Richard's work and looking at some of the other proteins involved. Can you have a cumulative effect in terms of that progression by targeting not only the amyloid, but tau and many other factors that are being researched across the world?
So, Mark, you talked about in your introduction about your work that you're looking at microglia. Microglia are talked a lot about at conferences. I don't think microglia probably make it into the news headlines. Tell us, what do microglia do? And what's the problem? What might be the challenge with microglia?
Yeah, so microglia cells are brain cells that are looking to, in essence, seek and destroy any kind of pathogens or any kind of toxic peptides that are presented to your brain. So they can be seen as the kind of the police force of your brain, surveilling your brain kind of 24 hours a day, 7 days a week, to keep an eye and make sure that that brain, the environment for those nerve cells, is optimal for them to perform. So they're really kind of seeking out and destroying anything that comes in that shouldn't really be there. So picking up on the kind of amyloid and the tau, these are proteins that shouldn't normally be there in terms of the actual accumulation and the forms that they take. So our microglial cells sense that, sense the danger, and start to try and then clear that danger. One of the challenges that microglia face is that the sort of tsunami wave that occurs that actually compromises their ability to start to clear some of those proteins that we know contribute to the disease. So in initial stages, there is, in essence, a good response from microglia. So they start and manage well to clear some of the, in essence, toxic proteins. But eventually the toxic proteins win out, and then actually the microglial cells turn from, in essence, not only good cops, but to bad cops. So they start to actually contribute to the disease process as well. And there's some really interesting research now highlighting that kind of detrimental role that microglial can pose as, in essence, the disease progresses.
So how do they turn bad cop? What is it? Do they end up just hanging around and filling the space, or what makes it bad?
Yeah, so normally their kind of physiological role in terms of kind of surveilling the brain is
I'm thinking Pac-Man here. Microglia is a Pac-Man. He's going around, he's munching up.
Yeah, that's exactly it. So they actually start to then, in essence, destroy some of the healthy neurons. So in essence, one of their roles in physiological or normal healthy brains is to prune some of those connections. So where connections between nerve cells aren't being used, microglia come around and then prune those unused, in essence, connections. But what you see in the disease process is that kind of regulation goes, in essence, awry, and they start to, in essence, engulf and destroy some of the neurons that you actually need in terms of being able to maintain those connections and brain activity.
So thinking about how we translate, what exactly is it that you're researching? Is it just what goes wrong, or is it, say, looking at how you might replace those faulty microglia, for example, or fix them? What is it?
Yeah.
Where does your work fit in?
Yeah, so we're looking at sort of some of the fundamental processes. So one of the things that microglia rely on is motility, so movement. They need to be able to be highly motile to respond to that danger. And you can look at that in the laboratory and look to see if you can find drugs or molecules that enhance that kind of motility, or in essence suppress that motility.
Like the power pill.
Yes, exactly. Yeah. We've got a theme going. We're going to have a bit of a trend.
I know, sorry.
Yeah, but exactly. So that's so in essence, we are looking at microglia in very kind of isolated conditions to see if you can modulate their behavior using some of the disease related peptides that Richard and I are investigating, and seeing if you can overcome those deficits just in one aspect of that particular microglial cell.
What do you think, Richard? How are you looking at the same thing? Or it's not the same at all, is it? I mean, how do you look at yours?
Well, yeah, so we're we're looking at how you can disrupt the tau aggregation pathway. So if you can...
So this is two-pronged, isn't it? Mark, what you're doing wouldn't stop those things coming in the first place. It'd just not necessarily the underlying cause as to what's making the amyloid. You just be able to fix it.
Yeah, so we'd be able to try. And so if Richard's in essence knocking tau out the park, we are helping the cells to try and in essence deal with any kind of leftover tau or kind of normal levels of tau.
I do understand this. I'm trying to think of this in the perspective, because a good majority of our watchers are coming on Facebook and YouTube who won't necessarily have the science background. So Richard, come on, sorry.
Yeah, no, absolutely. I think both aspects of our work will sort of work synergistically in a way. So neither would work without the other. So, if we can, as I said, tau aggregates within the nerve cells. And when that nerve cell dies, pathologic tau gets released into the next cell and so on. If you can prevent that tau seed, as we refer it to, getting into the next neuron, so we're looking at binding it with an antibody, or also within my funding company, they've been looking at a small molecule to do the same thing. Can you bind that, stop it getting into the next cell, and then systems in the brain, like the microglia there to protect from foreign bodies floating around, would come in and clear it away before it has the chance to cause any more damage further downstream. So certainly they're going to work hand in hand.
So I think people who are watching are going to have a little bit of a sense as to what's going on in the brain now. So just a question you weren't prepared for. So when you get news this week of this new anti-amyloid treatment, for example, this lecanemab, what's that doing? Where's that fit into this process of what you're both looking at, this drug? What's that drug doing? Does that mean that your work isn't necessarily, the microglia actually won't get broken if that's doing its job, or does this not stop it in the first place?
So I guess the kind of therapies that have been in the news recently are looking at the amyloid. So that's focusing on the amyloid peptide, and what they're looking to do is decrease the level. So it's an amyloid similar to maybe some of the technology that Richard's using to try and in essence attach and break up the kind of amyloid aggregating into those plaques.
Not using microglia, but using a different mechanism.
So it's not using microglia per se, it's using a kind of chemical approach, but the microglia are there to then mop up that kind of debris. So it doesn't kind of remove all the kind of surplus amyloid that's maybe aggregating. It's just breaking up into maybe more bite-sized chunks that the microglia can then manage, as opposed to kind of getting those big kind of aggregates that then go on to form the plaque. And then that's when the microglia start to struggle. So it retains the kind of microglia in a physiological sense, but they still need to be able to, in essence, manage those particular amyloid chunks that are being presented to them by the amyloid the antibody in terms of the therapy.
So when So
there's also other people that have, in essence, mutations in essence, genes that are encoded for microglia. So some of the, in essence, genome-wide association studies that kind of leads to our kind of fundamental research pinpoint some receptors on microglial cells. So it might depend on that individual in terms of their genetic background and why they're maybe more predisposed to the Alzheimer's disease to think about some of these targeted therapies. So we're almost thinking about kind of personalized medicine down the road in terms of what would be more appropriate for maybe a tau-based one or an amyloid-based one, or a predominantly more kind of global attempt.
So do you both are you working in dishes? Are you working in in mice, or in human post-mortem tissue? Where does this research happen? What's the medium for it? Mark, go on, you go first.
Yeah, so we are kind of across all different kind of species. So we look at human pluripotent stem cells. So they're the kind of best translatable element, but a lot of our sort of fundamental kind of first exposure to these kind of compounds is in mouse microglia. So starting to see what effects we can see and getting robust data sets before then translating into the human cells, because human cells are I'm a great advocate of using kind of stem cells, but it does increase the complexity based on the genetic background of those cells and the control experiments that you need to do to verify and also the heterogeneity. So us as a human population are very varied in terms of the cells that we might be looking at in a dish. So some of the responses that you'd expect to see might not be translatable across the whole population as a whole cohort. You might have to start to sub-select individuals to see your large effect that you've previously seen in maybe your mouse cells or your cultured cells.
How about you, Richard?
Sorry, mute button broken. So we start basically in the lab. We've developed what's called a tau antibody library, and to do that, the immune system has developed over millions of years to get really good at sort of protecting us from any pathogen that we come into contact with. And we've totally hijacked that process. So we injected a sheep with tau protein. The sheep recognizes this as a foreign body that shouldn't be there and produces antibodies against that. And we're able to isolate the peripheral blood lymphocytes from the sheep, which essentially contain the recipe for all of the antibodies that sheep is ever going to make. From that, we can isolate them in the lab, just in the test tube. We can take out the antibodies that have the specific properties that we're interested in. And by doing so in the test tube from there, we can look at how the antibodies we've isolated work at stopping tau sticking together. And that was very earlier on. So we took a panel of around 30 or so, and we narrowed down to one antibody that had the best properties that we were looking for. And we're ongoing studies looking at how that affects in transgenic mice. So mice that have been transgenically developed to produce human tau protein that goes to aggregate, how the antibodies we made can prevent that process.
Why a sheep? Do they make better antibodies?
They do, yeah. So one, they're big and relatively docile, so they're easy to catch. And
we shouldn't laugh. It's not funny.
It's but it's a really important thing when you're thinking about your sort of host animal. But for whatever reason, evolutionary-wise, sheep develop antibodies in a slightly different way to humans.
Genuinely didn't know that.
Well, yeah, there you go. There's a take-home.
Did you know that, Mark? No.
I was oblivious to the use of sheep. We use llamas, but that's maybe for a different reason. But sheep are.
So am I right in thinking then? So when a drug like this comes along that shows like it's got potential and there's some, this might start getting used, do you then need to start using samples that have had that drug in their system? Because you want to know if how your microglia are going to respond on amyloid that's had that drug treatment, that chemical. Is that something that down the line practically happens, or you would hope?
Yeah, so I guess that's kind of.
Am I jumping 10 years ahead?
No, I guess that's maybe what the clinical trials are going to tell us is like what the changes kind of long term are. These are kind of exciting results, but it's maybe what's the long-term effects of looking at sort of incubation or administration of these new medicines. And again, it might be looking at, what's the consequence of these? So it might change the microglial activity in a different way to what we anticipated further down the line. And again, so it's about understanding those, in essence, changes. And it might be that then we can then offer another medicine on top of the anti-amyloid therapy to then offset some of the negative side effects that we might see further down the line.
What we talk about in the dementia community, particularly that the public might not be aware of, is that there's a lot of likenesses drawn to things like early stages of HIV treatment and things like that, isn't it? That this is kind of how that ended up building up with these different elements.
Yeah, exactly. It's a good comparison to say that actually by chipping away, forming a little bit of the jigsaw, we can get that complete picture. And yes, it is a small change, but these changes actually make a difference in people's lives. So again, it will start to inform research going forward so we can get much better targeted and much more, in essence, better, bigger responses in terms of those effects that we're looking for.
And highlight the importance massive importance of collaboration between and where organizations, particularly, I know neither of you are in this, but organizations like the Dementia Research Institute are good at this in terms of bringing all different people together, because you want you two to be collaborating and working in the same space so you can share those things and have access to the data and the open science, open access to the data and the patients, and publishing negative results is so important so that we don't keep replicating the same thing. We're coming up to time, and I see my next guests already joined. So Mark and Rich, is there anything else you'd like to add before we wrap up? What's exciting you right now? Is there anything? What can we expect? What's keeping you on your toes?
I think it's a great time to be in the Alzheimer's research field. There's so much new and exciting data coming out, and it seems like we're getting incredibly close to breakthroughs that are actually going to really benefit patients. And we're going to hear from so many more people today. There's still so much good basic research going on in labs like my own and in Mark's that is going to just keep on rolling on. And yeah,
they really are. Thank you very much, Mark.
Yeah, just a quick sign off from me, just to say, I think the proof's in the pudding. Like fundamental research, all that research that's gone into the kind of amyloid story, gone into understanding how these molecules work, actually now makes a difference. It might have been a long time coming, but we're getting there, and that shows that the trajectory now should to push on from what we've learnt and keep going with the kind of basic research, because that informs kind of actual things that are tangible in people living with dementia.
Thank you so much, Dr. Mark Dallas, Dr. Richard Lofthouse, who have joined us from Aberdeen and Reading. Thank you so much for joining us. And if you want details on how to follow Mark and Richard and their work, you'll find those on the guests page of the chatathon.uk website. Thank you. And our next guests are already here. So that piece of paper goes in the bin. I'm gonna turn on the next caption, because coming up over the next half an hour at 1 o'clock, I'm going to give a quick reminder that if you're enjoying watching today, not only are we here to share some of the amazing research taking place, and over the next half an hour, we're going to be talking about data, omics, predicting risk and progression, but we're also here to raise money for brilliant charities. Alzheimer's Society, Alzheimer's Research UK, the Lewy Body Society, and Race Against Dementia all fund research in the UK, and they do an amazing job of it. So please do give what you can at chatathon.uk/donate. No matter how big or small, it'll make a big difference to us and to help keep funding the work of the people you're hearing from today. We're just going to play a quick film from one of our sponsors while I leave the room for a second.
Think about this for a minute. The brain has masterminded everything we've ever known, everything we've ever discovered, ever created, every idea that's ever been, everything we've ever felt, ever seen, ever heard, ever said, or dreamt. So when it comes to Alzheimer's disease getting the better of our brains, well, you can't outsmart the smartest thing in the universe. Which is why the brightest minds behind Alzheimer's Research UK are making incredible breakthroughs.
Alzheimer's doesn't stand a chance. We can all use our brains to do something amazing. Simply share this film and spread the hope.